Shortened or truncated genes caused by premature termination codons in mRNA can lead to a variety of genetic diseases. Fortunately, our cells have mechanisms to catch these early stop codons and degrade the mRNA before it is translated into proteins. However, this mechanism isn't perfect, which is why researchers study ways to help improve this process and improve people's health. On this episode of RNA Insights, we've talked with Dr. Kristian Baker, Associate Professor of Genetics and Genome Sciences at Case Western University and CEO of the RNA Society, about how nonsense-mediated mRNA decay impacts cells, as well as the ways that the RNA Society has created a community for RNA researchers of all levels. Welcome to RNA Insights. All right. Thanks so much for meeting with me today. Do you want to start by just kind of introducing yourself and saying who you are and what you do?
Hi, Grace. It's great to be here. My name is Kristian Baker. I'm an associate professor in the Department of Genetics and Genome Science at Case Western Reserve University in Cleveland, Ohio. I've been at Case for 20 years, running a research program focusing on understanding the molecular mechanisms of nonsense mediated mRNA decay, which we'll talk about more. And then I kind of have a side after hours role in leadership in the International RNA Society.
Cool. Well, thanks again for being here. To jump into it, how did you initially become interested in molecular biology and RNA research?
Oh, that's a great question. I think there's two seminal moments. In high school, when you branch out of science, a general science program, and you take physics and chemistry and biology. It was my chemistry class when we started talking about biochemistry and proteins and DNA and RNA, and that just really captivated me. It was really an exciting area, and so I started thinking about science differently than just a class, but I was really engaged. And then that drew me into undergrad research, or excuse me, undergrad studies in science. I'm actually a trained chemistry major, but I took some biochemistry classes, and it was a high level nucleic acid biochemistry class in the early '90s that really got me into molecular biology because it had a lab component where we started doing cloning, plasmid cloning, and real molecular biology, nucleic acid biochemistry, and molecular biology. And after that class, I started working with the professor and never looked back. I continued doing RNA research and gene regulation research since then.
Very cool. When you went into undergrad with this chem focus, this chemistry focus, did you expect to get more into biochemistry, or did you think you were going to go a different path with chem?
I liked chemistry and I liked the labs, but I think once I started to take the biochemistry classes and do more of the cloning and the molecular biology, I really enjoyed it. I actually did a 5-year undergrad because I ended up doing a double major in biology because they had more molecular biology classes. So I was on the fence, and I think that they're not independent. I actually really rely on my chemistry background when we do research in the lab. I mean, biochemistry is really an instrumental part of molecular biology and thinking about the experiments and troubleshooting, but really also how the cell regulates things. I mean, chemistry and biochemistry underlie all of those processes. So it took a little longer, but I kind of stepped on both sides of the fence, and I think it's really benefited my career.
As you mentioned in your intro, a lot of your research has to do with nonsense-mediated mRNA decay. Did you get into that fairly quickly after college studies, or did you start with something else in molecular biology to get there?
Oh, that's a great question. So, in undergrad, I studied gene regulation through RNA and RNA structural modulation of translation in bacteria. And that was a really cool kind of intro into RNA research. And I actually started my PhD studies doing work on alternative splicing of the cell determinant protein involved in cancer metastasis that is still being worked on today. But I had a bump in the road. My professor left the university, for another opportunity, and so I then moved to a different, type of research where I studied the interplay between mRNA translation and decay, and I did that in bacteria, and it's a really interesting system, and the interplay is tightly coordinated both in bacteria and in higher eukaryotes.
I've linked to Baker's first paper on the interplay between mRNA translation and decay in bacteria in the show notes. It's called Ectopic RNA sites promote bypass of 5' end-dependent mRNA decay in E. coli.
And that research led me to want to move into eukaryotic system and study again translation and RNA decay and how they're coupled and how they're regulated. And obviously, nonsense mediated decay is when you have a defect in translation and that impacts the turnover and the metabolism of the RNA.
Yeah. Do you want to elaborate on that a little bit more for our listeners, just to bring everybody up to speed before we start getting into the brass tacks a little bit?
Yeah, so it just depends on your background. I mean, nonsense media decay is really quality control. And I kind of think about it the same way you would do manufacturing. I mean, don't want to produce something that we didn't intend. So mRNAs are intended to produce one or more protein isoforms that are functional and useful for the cell. But if mistakes happen and that RNA is not translated completely to produce the full polypeptide, you get an aberrant or truncated polypeptide. And so the cell has this amazing, all eukaryotic cells have this amazing process where it can inspect RNA and determine whether it is going to be functional. And if it isn't, then they remove that from production. So again, very much like a manufacturing process where you do quality control to inspect and remove, the cell does that. And in this case, it can recognize, for the most part, RNAs that are not translated completely due to a premature stop codon.
For your research into this mechanism, were you studying more how this mechanism happens or looking at fine tuning what occurs? What did your research specifically look like in your lab?
So I think from the beginning we've taken two approaches, and then it goes back to that manufacturing analogy. First of all, how do you detect? How does the cell? It's one thing when you're standing on a manufacturing plant and you can see a defective widget, for example, or you weigh it and you know that the material is not right. I mean, there are different assays for that. How does the cell assay whether a transcript, an RNA, can produce a full length protein or a truncated protein? And I think it's important to keep in mind that that RNA can differ only by one nucleotide, right? A single nucleotide can introduce or change a sense codon into a nonsense codon or a stop codon. So, with such a small change to the RNA, how does the cell inspect and recognize? So that's been one focus of our research is understanding kind of substrate recognition. How does the cell see that? And then I think the other big part and what is a more of a focus now is understanding once that recognition happens, how does the cell then lead to a cascade of events that ultimately leads to the rapid degradation of the RNA? Because again, just like the manufacturing analogy, if you don't take the defective part off of the conveyor belt, it's going to continue on. Even though you recognize it, you need to remove it. And in karyotic cells, those transcripts are removed by rapidly degrading them. So preventing them from producing or accumulating any of these polypeptides that are short and not functional.
Say that the defect isn't removed from the conveyor belt. If these quality controls don't happen, how does that impact cells or entire organisms?
Well, we know that NMD, the process of nonsense mediated decay, is absolutely essential in mammalian cells, right? It's embryonic lethal in mice and mutations in the machinery that's responsible for NMD in humans lead to different levels of intellectual disability and autism. So this process is really critical part of gene regulation and maintaining gene regulation fidelity. So we know that that's important. If it doesn't happen, say, on an individual transcript level, so if you fail to remove and you start accumulating these polypeptides, depending on the protein, the polypeptide that's produced, it can be dominant negative or it can be unfunctional. It just really depends on where that stop codon is and what the truncated polypeptide can produce. So it's really important, and it's important to clean up a whole bunch of mistakes that happen in the cell. When we're taught biology or gene regulation, we think that the cell is pretty efficient at all of these processes when you do gene regulation from DNA to RNA to protein. But I think in the era of RNA sequencing and high throughput analysis of transcriptomes, we recognize that there is a lot of variability in kind of the processing processes. And so a lot of alternative transcripts and a lot of different mRNAs that are non-functional. And so NMD, it turns out, helps clean up a lot of those unintended transcripts that happen just from plasticity and errors in during processing. So it's not just the ones that have mutations, say in the DNA, but also all of these transcripts that end up having premature stops because of errors in processing or other events during gene regulation.
So, to recap, NMD occurs when a cell inspects a translated mRNA and finds that it will produce a truncated or shortened polypeptide. This decay occurs in all eukaryotic cells, but cells' mechanisms aren't perfect. Mutations can occur when cells let truncated polypeptides through their inspection. That makes sense. At your lab, I know that you have uncovered, like you just mentioned, some of these mechanisms of when M&D is occurring. How did some of those discoveries feel over your time? Because obviously you've been at your lab for a while, and like you mentioned, the technology, the RNA research has changed so much, even just in the past 5 years. How has those changes impacted your lab or what you had found?
That's a great question. I think the fundamental questions we're asking about recognition and then what happens later to initiate degradation of the RNA, that cascade of events, that signal transduction after, those haven't changed, but maybe some of the tools we use, we've adopted new technologies and new tools to ask questions in a different way. I think we've been successful at combining both kind of some of the traditional molecular biology tools using reporters and making very simple changes and asking about the effect on whether that transcript is recognized. And then couple that with high throughput like RNA-seq and ribosome profiling. We do clip analysis to understand how our proteins involved in NMD binds RNA and recognize substrates. And we moved into proteomics, which is a little bit of a new area for me, but we're learning a lot about those interactions. And again, that's to tease out those events that happen afterwards and try and get a dynamic picture of how the RNA is then degraded. My philosophy is that if you want to target this as a therapeutic, this process, we need to understand it at a very complete molecular level. And that includes all of the interactions that are going between the nonsense-mediated decay machinery and the ribosome and the RNA and the decay machinery. It's a complex ecosystem. And so we need to understand all of those interactions.
For sure. That leads into my next question well, which I was gonna tie in. Obviously, this podcast talks a lot about RNA therapeutics. Where are we in that stage, or where are you rather in that stage of looking at these interactions and seeing how this ecosystem works in terms of be able to identify targets, RNA therapeutics?
So we're using proximity labeling and we're using other approaches in vivo. I like to do in vivo analyses rather than in vitro biochemistry because our system's quite complex and there's we're just on the forefront of trying the field is on the forefront of trying to reconstruct decay and translation and NMD in a tube. We're not quite there yet. So we still take an in vivo approach, and so we've adopted proximity labeling to try and understand the protein interactions that are going on.
Proximity labeling allows researchers to map interactions between cells and their environment. This method uses an enzyme that binds to a protein or RNA sequence of interest and tags a small radius around this binding site, showing where the interaction is occurring. I've linked to a review of in vivo proximity labeling in the show notes called In Vivo Compatible Proximity Labeling Technologies.
And then we've taken that a step further and we look at different mutants that we had in the nonsense mediated decay pathway and try and then piece together the interactions that happen between the different mutants and because they're stalled at different processes. So that's what we're doing for the interactions. And then obviously we use a yeast system still to study the mechanism of NMD. And the benefit of the yeast system is, once we have some interesting data from our proximity labeling, And coupled with work with collaborators and others in the field on IP analysis and protein interaction analysis, um, we can easily test the importance of those interactions. We can make a lot of different yeast strains very, very quickly and analyze whether those protein interactions are actually key to events in the cell.
Yeast is common for researching NMD because yeast has conserved the NMD system and it grows incredibly quickly. I've linked to one of Baker's papers using yeast in the show notes called Analysis of Nonsense-Mediated mRNA Decay in Saccharomyces cerevisiae. Do you expect most of the interactions to be really vital and really important for the cell? That's interesting to think about. I guess I never thought about the idea of that some are more important than others. That makes total sense. How did that spread look?
Yeah, it's interesting. We've known of interactions between the NMD machinery and several factors that are key and several factors that are newer for a while, right? From yeast to hybrid experiments, from protein interaction experiments, from our proximity labeling experiments. Some of them don't seem to have huge impacts on whether a substrate can be recognized or degraded. So that would suggest that those interactions aren't important, or maybe we're not assaying them in the right way. So I think it's important to keep an open mind, to assay and try and understand those interactions. But then if they're conserved and we keep seeing them over and over again, we might be overlooking something. On the other side, I mean, guilty by association. Just because they're interacting, that doesn't mean that they're in cahoots in mediating this process. So it's something that you have to untease and figure out by doing kind of in fundamental molecular biology and genetic assays.
I feel like that's interesting for a therapeutic standpoint as well, with deciding what to target if unsure, still figuring out the interactions. It'll be interesting to see in the future. But speaking of the future, obviously right now you're looking at these interactions. What would be the next steps of this to get therapeutics to the clinic? What would that process look like from where you are right now to getting to patients? Obviously, that takes a long time, but
Well, there's some exciting developments, with there's a almost 25 to 30% of single allelic genetic diseases have a nonsense mutation as the underlying cause. And that's just not one, that's any genetic disease.
single allelic genetic diseases occur when a mutation of a single allele of a gene can cause a disease, such as Huntington's disease. These diseases occur when a mutation has a dominant inheritance pattern. You can learn more about single allelic and single gene genetic diseases in the show notes.
And so there's a lot of interest in being able to figure out how to do one of two things: convince the cell not to stop at the premature stop codon and read through to the normal stop codon. So that's nonsense suppression, suppressing the recognition of the nonsense mutation. And that's really convincing the ribosome not to stop at the premature stop codon. And then the other approach is trying to inhibit the degradation of these RNAs in cases where the stop codon is late enough in the message and the truncated polypeptide might have some functional significance. So even though it's not the full length peptide, it still would have some function if it were to accumulate. So in order to get accumulation, we'd have to inhibit the nonsense-mediated decay, the second step, right? The targeting of the RNA. So the two approaches are, a lot of people are thinking about this. There's some really nice work that just came out of the David Liu lab at Harvard, looking at tRNA suppressors and suppressing nonsense mutations using CRISPR, prime editing.
I've linked to this paper called Prime Editing Installed Suppressor tRNAs for Disease Agnostic Genome Editing in the show notes.
I think there's other approaches, again, trying to inhibit NMD. The problem being is we would like to do that on a message specific basis, right? So a disease like cystic fibrosis, where you have a number of mutations that are caused by nonsense mutations, you'd only like to target the CFTR mRNA and not all of the NMD processes going on and inspection and the degradation of other messages. And so I think if you ask me, the challenge will be to get that specificity. I do worry, although some of the data looks good, I do worry about doing not general nonsense suppression. And I do worry about inhibiting NMD in cells completely inhibiting the NMD machinery, because that will have huge consequences on the transcriptome.
Some recent research, such as nonsense mediated mRNA decay inhibition reshapes the cancer immunopeptidome, suggests that blocking NMD can activate T cells and potentially increase tumor immunogenicity. However, as Baker suggested, NMD inhibition could also allow truncated proteins to be expressed, negatively impacting how cells function. Yeah. No, it's so interesting. Everything is so interconnected. The space that we're in right now is very interesting to see how it will continue to grow and develop.
It's
Um,
an exciting time for sure.
I'm going to pivot here in a second to your other role that you're mentioning and working with RNA Society, and I know the meeting just occurred. But first, to continue with your research, what is the most challenging part of your research?
I think I have a great group, but ideas are easy. It's always you have more ideas than time or capacity or grant funding. We all wish the research would move forward faster. We could ask more questions. We could get data faster. I think that's always a limitation. And then we're in a challenging funding environment these days, especially in the United States and especially for RNA research. I've been fortunate. We have funding. I've been fortunate to be funded by both the NIH and the NSF, but it's a challenging time. And so, really, I think many of my colleagues would agree that just getting funding, being supported for more foundational research, really understanding mechanism and foundational gene regulation is getting more difficult and in this environment. So that certainly is a challenge.
Um, Imagine. I ask every podcast person this question. The answer tends to be fairly similar. On the flip side, though, what is your favorite part about doing your research?
Oh, I love training scientists. I love being in the lab and talking to my graduate students and my postdocs and my research assistants about be scientists thinking about data. What is this experiment? What does the data mean? What can we learn from it? What do we have to be cautious about? Over interpreting. How do we think about science, right? And the core of science. And you can teach a little bit of that in the classroom. And I like teaching, but it's not until you're in the lab and you're looking at data and you're thinking about ways to ask a question and frame a question. That's just great. That for me is makes my day. And when you see that transition, especially with graduate students, from kind of relying on you to taking that leap to being able to make those connections and those interpretations and deciding what the next step is. That's just the most rewarding part of being, I think, a PI.
That's awesome. That's so fun. Pivoting here then to what I talked about earlier with your other role, I know you are the CEO of the RNA Society, and the annual meeting just happened a couple weeks ago. Do you mind first just talking about the society in general and what it is and what you all do?
Okay. The International RNA Society is an international organization for RNA researchers and educators. We provide a community and for researchers and educators to come together. We have some benefits for being a member, but I think foundationally, we provide an annual meeting where we can share the science for research, the science that comes out of mostly academic labs, although we're partnering more with industry and trying to bring scientific RNA research developments in from both the industry side and the academic side, and RNA education. So, when the RNA Society was founded, it was to provide researchers that opportunity to come together and share and collaborate and network on their research discoveries and to move RNA research forward. And then we also have a scientific journal called the RNA Journal, sponsored by the RNA Society. And that again was provided and established when the RNA Society was established to provide a place for our members and non members to publish their research. High quality publication that's edited by scientific researchers that have labs all across the world that are your colleagues and provide you a place to publish your research and get peer review and move the science of RNA forward.
I've linked to the RNA Society journal in the show notes. That sounds like a great community, a great space. When did you get involved with the society, and how has your progression and your experience with it been?
Huh. The society was a community for me even as a graduate student. So I went to my first RNA Society meeting when I was doing my PhD studies, and the community was fantastic. I learned so much about RNA. RNA is so broad. The RNA science is so broad. I was overwhelmed with what I didn't know. I was so excited about all that was going on, and the community was just so welcoming. It was just a great community. So it influenced me early on in my training. And then when I came to Case Western Reserve University as a faculty member, I hadn't appreciated the history that between the RNA Society and Case Western. So the founding editor in chief and of the RNA Journal was the director of the RNA Center who hired me here, Tim Nielsen. He was the editor of RNA for almost 30 years until 2022. One of the faculty members was also served as the chief financial officer of the RNA Society. So there was a long history of the RNA Society having a presence at Case Western and in our department. And after having such a positive influence with the community, there was an opportunity to serve as the chair of the membership committee, which I stepped into in 2014. And I did that until 2019.
Baker is currently the chief executive officer of the RNA Society.
So that's how I kind of got roped in. But I just felt it was just such a community that I wanted to be part of. If I'm going to serve, I really wanted to serve the community in this capacity.
How has the community and the society changed over time from when you were a grad student to now?
I mean, I think it's gotten bigger. We have more members, there's more benefits, there's more people involved in the leadership trying to provide what scientists need, RNA educators need now to do their work and to promote education and research. I think there's a fundamental tenet, though, that is exactly the same. One thing that I noticed when I was a graduate student, and it's a priority now, including our meeting in Montreal that we had 2 weeks ago, is that we focus on trainees. We focus on graduate students, postdocs, now undergraduate and even high school students have come to our meeting, and early career researchers who are just becoming independent. The focus is to try and help them, whether it be networking or, again, providing them opportunities to present their work at our annual meeting, any career development opportunities. So the society has always been focused on the trainees and trying to make sure that the trainees have opportunities, a community, a network, and support for their careers. And that hasn't changed. And that's just such an important part of the society.
For sure. That's awesome. You mentioned this year's annual meeting in Montreal a couple weeks ago. Can you tell me a little bit about the meeting this year and how it was and the new breakthroughs in research or just some highlights perhaps?
Oh, wow, that's hard. So, our meeting was in Montreal and in Canada. It ran from May 26th to the 31st. We had over 1,000 researchers from all over the world come. We had a great contingent from Canada. We had almost 200 researchers from Canada, and we had 175 oral presentations. So, I can't summarize those, including several concurrent. So, you can't be in three places at the same time, but really a lot of excitement for the format this year. The Montreal Congress Center was an excellent facility for us. It kept us together. There was just a lot of networking. I saw students with their posters on the ground after the poster presentation talking to people because there wasn't enough time. I think there was just a lot of excitement about this year's meeting and what we're seeing with the collaboration and the movements. There's still a lot of work on foundational RNA research, but there's really been a transition, I think, in the last maybe since just before COVID to understanding how RNA fits in disease, in human disease, and how we can maybe target how our understanding of fundamental RNA biology can be overlaid now in how we're understanding mutations and disease and the role of RNA in contributing to that disease. So there's just a lot of excitement there, and you could just feel it in Montreal.
Cool. Oh, that's so fun. If someone wanted to find out more information about the society and like previous meetings or next year's annual meeting, where would you recommend for them to look?
The RNA Society website is a great place for all of that information, and it highlights not just our annual meeting and our journal. We have lots of programs going on. As I mentioned, we've expanded. We have 8 different standing committees, all serving our members in different ways, whether it's awards or our social media or a journal. We have all sorts of things going on that's on our website. We just relaunched a new version of our website. It's really great. It's a lot cleaner and easier to navigate. And right on the front page is our information about our meeting next year, which is in early June in Strasbourg, France. So we will be returning to Europe for the first time in 3 years. We were in Edinburgh in 2023. And it also hints at the board of directors has just approved that our meeting will in 2028 will be held in Sydney, Australia. So we're very excited to return to Asia Oceania, and it'll be the first time that our meeting has been held in Australia. And they are so excited about hosting us. And what we need to do is just make sure, lower the barriers and make sure that our members can get to Sydney, because I think it's going to be a fantastic meeting too. So all that information is on the website, and take a look. And you can always contact me if you have questions or want more information.
Sounds good. I'll be sure to link to the website in the show notes of this episode so people will be able to access it very easily. And I did. You can access the link in the show notes. Pivoting back to you, how has balancing your research and working with the society, how do you balance these roles? I know at the beginning of the episode you mentioned kind of having the night shift and the day shift and whatnot. How do you balance it all?
Oh, I love that question because I've asked lots of people. I have wonderful mentors and that I've had throughout my career, and a lot of them have been women that I have just admired because they're balancing family and career and maybe additional positions like editor positions. And it's a balancing act that changes every day depending on the priorities. Um, yeah, it's hard, but you do it because you love it. I mean, again, the RNA Society work is something I really enjoy doing, and if you're gonna do service and additional work, you have to really enjoy it. So, the community's been really good, and we have a great group of, leaders and committee chairs that we work together, and we all understand that we have full-time jobs, and that we do our best to balance when we have grants due or childcare or something else. So I think just being open about the challenges and all of the things that we have to do, and again, this community being accepting of those challenges has just allowed it to happen. People step in and support each other, and that's really helped for me. It's a little bit more work than I had imagined, but partially we just have such momentum that it gets bigger and bigger every day, but it's such a fun it's really has changed my career. I'm a scientific researcher, but this aspect has really fulfilled, something that was is a nice addition to the science.
Thanks again to Dr. Kristian Baker for joining this episode of RNA Insights. Before we leave, here are a few pieces of RNA therapeutics industry news. Moderna announced that the US FDA unanimously voted that the benefits of mRNA 1010, an investigational seasonal influenza vaccine, outweigh its risks in preventing influenza in adults both 50 to 64 years and 65 years and over. Moderna has a US FDA Prescription Drug User Free Act goal date of August 5th, 2026. City Therapeutics announced it completed a $99.5 million Series B financing for its RNA interference therapeutics. The company recently began a phase 1 trial of CityFXI, an investigational RNAi therapy for thromboembolic diseases. Novartis presented data on Rapsido, a BTK inhibitor to treat chronic inducible urticaria. The phase 3 trial suggests that Rapsido reduces highs for people living with the disorder. Thanks again for listening to RNA Insights. We'll see you next month when we talk to another RNA expert.